Distinguished guests, dear colleagues, ladies and gentlemen, I would like to start by thanking the organizers of this 10th International Symposium of Rice Functional Genomics for the invitation to have an opening address today. It is indeed a great pleasure and an honor to be with you here and share some ideas of rice genetic improvements in the future. I would also like to congratulate the organizers of the theme or the topic of this Congress, driving the next generation of green revolution. I think that's put exactly to the point of what we should do, considering of the challenge what we have ahead in the coming decades. Along those lines, in my presentation, I would like to share with you what is the challenge. I think everybody in the hall knows it pretty much exactly what we need to do in the coming decades. But to summarize, and also to share with you some of the thoughts and ideas and plans of how we can cope, how we can respond to those challenges. So let's go ahead. So rice is life, as mentioned earlier already this morning. Half of the world population is dependent on rice. A little bit more than 100 million farmers produce that rice for the half of the population. There seems to be no slowdown in the consumption of rice. Exactly the opposite. The need and demand for rice is going to continue. Obviously, Asia is the area where rice traditionally has been the most important staple. But if we look at the other parts of the world, and especially sub-Saharan Africa, we can say that rice is the fastest growing staple, also in that very poor part of the world. And importantly, 40% of the consumption of that part of Africa is imported. So obviously there is a need to increase rice production also there locally. Rice price has a direct impact to food security and poverty. In 80s, 90s, till the early 2000s, rice price was a very much stable and a pretty low level. However, what we have seen after that is a slow but continuous increase in rice price. Actually, today we are almost at the level of double what it was in early 2000. 2008, we have the peak in rice price, when the rice price went up to the $900 per ton, and gave us a very direct indication what will happen if rice price gets higher. The number of people in Hungary came up pretty fast, gave us an indication what should not happen. Obviously, this also gave us a good opportunity to study out there in the villages, in the poor areas, how farms, how poor families cope with this sort of situation. A lot of important information is coming to teach us how we should handle this sort of situation in the future. In 2035, towards 2035, the world population will be around 8.5 billion. And the need for extra rice grows in the way as shown in this figure. As estimated by the models of Eero, we will need some 100 million ton more rice towards 2035. meaning a growth of 1.5% per year, or 8 to 10 million tons per year. Importantly also, as said earlier and shown is this figure, the need in not only in Asia, which is going to be the major demand, the need also in Africa is going to grow substantially. To make the things worse, climate change is going to change the environment where rice is produced. We can say that climate change will hit hard rice production in Asia, in Africa. Basically there where rain-fed rice is dominating, but also, of course, having influences to irrigated rice. There are many scenarios of how climate will change. What are the main factors? What is the main speed? When it will happen? However, there is also quite an universal consensus of the main things which will happen. Temperature will increase. How much and with speed, that's a question, but there is no doubt that will happen. There will be change in rainfall. And unfortunately, it looks, according the projections, that in many areas of rice production, rainfall will decrease, meaning a big challenge for rice production and particularly for rain-fed rice. Sea level will rise as a follow-up of temperature increase. This is quite an important thing, noticing that about 50% of the rice production increase during the last 25 years has came from the delta countries. Obviously, in these deltas, when sea level rises, the question of salinity tolerance will be a big thing. A lot of uncertainties, a lot of big changes in better, unpredictable things, more storms. That's also one thing which has been predicted in most of those scenarios. Climate change consequences. Rice system will experience more drought, submergence, salinity, heat waves. And we can also add to this list, although we don't know at the moment exactly how it will happen, but probably the pattern of biotic stresses, diseases, pests will also change. Perhaps the priorities of different diseases will change. Perhaps we will see a new emerging diseases. This is an area where our pathologists are doing very, very important work to see what might happen. Changes of climate change. At Eero, we think that those changes are exactly of those which the world's poorest farmers are facing today already. So indeed, what we do to improve their rice production and their lives through rice science is preparing for the future of climate change. Targeting is very important in science like genetics or breeding, where the product development takes quite some time, 5-10 years, perhaps even more. So we need to know what are the conditions and environments, what for we are doing our product development. Very important research is going in this area to help us targeting of our efforts. I show some of the slides coming from the CRISP research concerning of how these major climate change factors will influence rice production now and in the future. For instance, here, daytime, nighttime temperatures, where the risk areas are. Similarly, drought. What the current models say where the areas of drought are today. Flooding, the same thing. And very importantly, as we know that if you take a look to rice and compare it to the other crops, farmers tend to keep the varieties they have been using tens of years. And if we analyze often why it's like that, why they don't change to the more modern, higher yielding varieties, it's the question of quality. If the quality is not there, the farmers tend to keep those varieties they like to grow and like to eat. So quite a bit of work is going on to understand what sort of quality preferences we have in different parts of the world of rice production. Here just one example of the amylose content. This is quite a challenging work since we need to go down there and to really to study with the local people of why they prefer a certain qualities and why they don't accept a certain others. The maps I showed are about the current situation, the situation of today. What is very important here is that the research focusing on this area are soon ready to give also the predictions of how the different stress areas will change according and along the climate change. There are quite an emphasis also to try to make models to predict biotic stresses, which is of course very much more challenging than the abiotic stress analysis. There is no doubt that rice science, rice genetics and rice breeding has made a tremendous impact to the food security since 1960s. I just pick up a few things where just to show what a one gene can make. As we all know, the semi-dwarf or dwarf variety is introduced by Eero in 60s and a bit later, really changed the production level of rice, making available varieties which were able to use the higher impact level. Basically, those varieties doubled the rice production in rice producing countries. SD1 was the gene which was involved in most of the cases. There was also other semi-dwarf and dwarf genes, but this was the most important. Then came the variety IR36, which to our knowledge is the variety which has been cultivated the most widely compared to whatever crop on the globe, 11 million hectares annually between 1980 and 1989. And why it was so successful, of course, it was a semi-dwarf, but also having a good package of biotic stress tolerance. Early maturity is still a very important thing in some production areas where really to increase the production is to make 3 crops per year. Maturity level of 80 days, 90 days was the key to make it happen. And today we can see that in many parts of the world, in Southeast Asia, Sub-Saharan Africa, there is a demand of this sort of varieties again. To have a 3 rice or 2 rice plus another crop. But anyhow, 3 crops per year. Hybrid rice, male sterility, the wild apportion CMS gene, today used in about 90% of rice hybrids outside of China. And the latest one, abiotic stress tolerance, I pick up the SUB1 gene, which is now delivered to the farmers in South Asia, India, Bangladesh, Nepal, which protects the rice crop for flash flooding, submergence for about 2 weeks, sometimes even longer, if the quality of the water is good. I just came back a week ago from East India to see how these varieties are doing there in Orissa, Bihar, the poorest part of the world. And it was absolutely fantastic to see what is happening there. Varieties which they have been using the last 25 years or so, traditional tall, which in some level can tolerate flooding, which is, as they told, happening every year, sometimes 2-3 times per season, sometimes combined with drought. Now they are changing from those old varieties, producing some perhaps 1,000 kilo per hectare, to sub-1 varieties having 3-4 times more yield. It has been just amazing to see how quickly these varieties spread into the poor farmers. We started there 2009, and now 3.5 million, that's our estimate, 3.5 million farmers are using Swarna sub-1. Swarna is the variety the most popular in east part of India, in Bangladesh, cultivated with some 6 million hectares today. And the prediction is that Swarna sub-1 will completely replace the old Swarna. Few ideas where we should focus. I think this is quite a bit of question about prioritizing. We have limited time to respond, so we cannot do everything, but we need to focus on some core areas. Obviously, stress tolerance is one of those. To focus on those key factors which are already there, but which are getting even worse along the climate change. Flood tolerance, clearly the one. There are different traits under this title. Anaerobic germination, to be a rice plant or a rice seed which is able to germinate in an anaerobic condition. I think this is going to be one of the major things, because we need to realize that the rice production system is also going to change from transplanting to direct seeding. And that's because of the lack of labor in those countries. The labor in the villages who have traditionally helped farmers to do transplanting, they are moving to cities. And there certainly will be a lack of labor, and it's already there. So we estimate that change from transplanting to direct seeding is the major change of rice production happening in the coming decade, or perhaps even more. Flash flooding, submergence. We have sub-1, which is making a tremendous difference. We need to find more new genes which are additive to sub-1, or perhaps making the recover after flooding better than it's today. In many cases, after flash flooding, the water remains in the field, 20-50 centimeters, and that's a big stress for the crop. We have estimated that the area for stagnant flooding is at least the same as for submergence. A lot of work is going on also in finding germplasm and genes to tackle the stagnant water. Drought, as mentioned, drought and flood, they are kind of a sister and brother. Same areas, even within same seasons, these stresses then can go back to back. Rice seems to be different if we compare rice to other major crops. What the research now indicates that we can find big QTLs, and later of course genes as well, which improves drought tolerance of rice tremendously. At Eero, the team has identified today five big QTLs, working in a different backgrounds, quite a many backgrounds tested. They are stable in different environments. They work in upland and lowland conditions. What is the good news here is that some of those seems to be additive, giving us a chance to pyramide and to get a better drought tolerance in that way. Salinity tolerance, as mentioned, the delta areas and also in other areas. Very, very important. A lot of work done to identify new donors, and as this map indicates, it looks good. As in the case of trout, it looks also here that some of these QDLs and genes are additive, giving us a good promise that we can tackle this issue pretty well. Biotic stress tolerance, it's a kind of, I would say, the most challenging part of the story. Technically, it's pretty straightforward to introduce these genes to our elite backgrounds. But the challenge is, as everyone knows, is that the enemy, the disease or pest, is evolving according to lines of what we introduce to them. So how to make sustainable and durable resistance, that is going to be the problem. We think that a wide spectrum durable resistances can be found especially from the wild forms, wild species of Oryza. Here is just one example of blast genes identified from the other Oryza species. And the same type of research is going on also to find genes against plant hoppers and others. We think to enhance the yield potential of rice. We have since 80s, 90s, the yield potential of rice has stayed quite a same level. But obviously, if the stress tolerance is the key thing for rain-fed rice, we need to find ways to improve the yield potential of irrigated rice. Noticing that still the major part of the global rice production comes from irrigated rice. About 70% of the production, about 50% of the area. I just want to share with you a slide which was shown by Professor Longbing Yuan in last September in the International Hybrid Rice Symposium in Hyderabad, where the Chinese hybrid breeding is going. And you all know that Professor Yuan is the father of hybrid rice, as he is called. So we started with the semi-dwarf types, and now the trend is towards a bigger biomass crops. And their vision is a hybrid rice of 2 meters tall, giving an estimated yield of 18 tons per hectare. Where the challenge is here is to keep the harvest index at a high level, at the 0.5. If they are able to do that, the estimated yield is as indicated here. But also showing that the plant type, what they actually already have, the level of 12 to 14 tons, it's giving us a pretty good indication what we should do. I think that's the core thing of the yield potential breeding. Instead of looking the sink size, seed size, number of grains per panicle, number of tillers, we need to go and also to see the source size of the plant potential. Anyhow, again, rice has a very interesting genes for improving the sink capacity, and just to give one slide to show that we have started a project, Crisp New Frontier project, to focus on increasing the yield potential of rice by pyramiding the genes which our colleagues in Japan has identified and cloned. We need to focus on rice genetic resources. We just cannot overestimate the value of that, especially in the case of rice. If we look at rice gene pool and compare to the other major crops like corn, soy, or barley or wheat, we have just tremendous diversity within rice gene pool. 24 species, and as an outcome of the excellent research which has been done since several decades already, we basically can introduce a gene even from the tertiary pool of rice to the cultivated pool. There's more than 100,000 samples in the gene bank of Eero, copied to a couple of other gene banks in US and one in the Svalbard, Norway. And we have just touched a small part of that diversity which is in there. Certainly, I would say close to the number one issue of how we could respond to the great challenge of rice genetic improvement. As mentioned, wild rices are extremely useful for improving rice sativa. And just to indicate a few of the things which are ongoing, obviously abiotic stress tolerance, but even yield potential. Rice yield growth rate has slowed down, as mentioned earlier. And if we compare what is going on in rice and compare rice to other major crops, particularly the temperate cereals, we are a little bit behind on what is happening there. If we take a look at what is going on in barley or corn or wheat, the yield gain is much higher than what it's in rice. That's probably one reason might be that in those crops the private industry is driving quite a bit of the breeding and has been able to integrate the most modern technologies to make the genetic gain higher. That's just something we have to change. And I just would like to emphasize that certainly genetic gain of quantitative traits, meaning basically yield, is something we need to focus on, and we have the tools to focus on. To use the same approaches as used by private breeding industry for the temperate cereals and other major crops to go toward genomic selection. We need to speed up the breeding cycle from the crossing block back to the crossing block, so to really use the best parental materials faster than we do today. We need to try to find ways to shorten the breeding scheme. Doublet haploids, single seed descent lines, etc., which are routinely used almost as the only breeding method of barley or wheat. We need to find ways to do the same. We need to take into use the most modern quantitative tools, genomically estimated breeding values. All is ready there, and we have the data to do it. We just need to start to make the change. And very importantly, these tools, of course, make it possible for a program to monitor all the time, after every breeding cycle, what is the genetic variation in my breeding pool at the moment. Variation needs to be there. The challenge is to get the high average and high variance, and when it's there, the program will certainly deliver. Last point. The challenge ahead is big. There is no way that any single institution can do it alone. We need to mobilize the global rice science community to come together and hit the challenge. And that's exactly what the Global Rice Science Partnership, starting in 2010, is doing. Getting together rice scientists all over the world to work together and make our best effort to win the challenge. Today, CRISP is already accommodating hundreds of international and national partners. And we think this is really the portal or home for all research activities focusing on rice science. Within CRISP, just to give a couple of examples, there are excellent new initiatives to focus on the big things which nobody can do alone. Global Rice Phenotyping Network, which just had its second annual workshop last week at Eero, is one of the examples. Clearly, phenotyping is going to be the thing. Genotyping data getting cheaper and cheaper, new methodologies, technologies coming almost every year, and that part is, I would say, well in control. But how we do high quality phenotyping in different environments, focusing on the difficult traits, we need a global network. And that's what a Global Rice Phenotyping Networks is going to do. Very good start, and we are looking forward to get an excellent results out of this. Public-private partnership. We all know that private sector is coming to rice with a speed, I would say, never seen in any other crops. So this gives us an opportunity to work together with privates, and through that, having both public and private channels to make the impact at the level of poor farms. There is certainly great possibilities here. We started Hybrid Rice Development Consortium in 2008 to get together both public and private hybrid rice breeding institutions and companies. Today, this consortium have some 60 members, of which half are private companies. And just to show, the number of germplasm we have at Eero shared with both public and private institutions, it looks that, and it's not only look, but it's clear this is something needed. It has been growing, the material, it changed dramatically. Global gene discovery effort. I'm sure you will discuss of this issue here in the days to come. And clearly, we need something the same, a consortium, global consortium to tackle this issue together. Again, we think the Global Rice Science Partnership is the host of getting all of you together to work in trust, to get all the best talent, all the data together, and make the big impact what is waiting there and what is the promise there in these new technologies. I'm sure during the days you will change a lot of ideas of how to make the consortium, but clearly what I think is the right direction is to find ways to incorporate these activities into the CRISP. Just to summarize, I think there are three things important if we take a look to the challenge and what we can do. First of all, we need to prioritize of our activities, to focus on traits, genes, which really make the difference at the farm level, at the lives of those who are dependent on rice. So prioritize. We need to move fast. Time is against us. We need to move fast, meaning we need to exploit the most modern technologies to speed up the product development of rice. That's the second point. And the third point is we need to work together in a partnership. The challenge is too big for anybody else, any one of us to do it alone, but we need a partnership. Thank you very much.